All right.
This week in evolution, it's episode number 68, recorded on May 26th, 2021. I'm Vincent Racaniello, and joining me today from Salt Lake City, Nels Eldy.
Hey, Vincent. Good to be with you. Also called Small Lake City, as I think you were starting there.
Yeah, I can't speak properly, you know. I have a problem speaking and moving two hands to do things here. I'll get used to it, I guess, eventually.
Oh yeah, this live streaming, it's sort of like driving a car with a manual transmission. You're pushing in the clutch, pulling the brake, shifting gears.
Which I do every day. I should have no problem doing it. Salt Lake City. How are things in Salt Lake City? Is the weather getting warmer?
Yeah, things are good here. So moving into early summer, we're dry, extreme drought conditions, but thankfully some thunderstorms had moved through in the last 2 days, greened things up here. Also progressing, on the SARS-2 front. So the university, for example, where I'm joining you from LD Lab Studios, moved from a restricted research posture with basically very little in-person lab time to a modified restriction. Still trying to definitely limit our in-person time, our collisions, but starting to feel a little bit of that optimism that things are moving forward really well with a vaccine and in other ways. How about there in New York? What's the latest from there?
Things are getting almost back to normal. I think everything is slowly opening. It's still restricted numbers, but restaurants and all that stuff are open, bars, and by July 1, I think full open.
Wow! Yep.
And I still wear a mask when I go outside, but the traffic is back. I have traffic now in the morning when I drive in. So.
Yeah, I hear you. Definitely a sea change, right, from even a few months ago.
For sure.
Yeah.
But it's good. It's all good. 50% vaccinated today, right? The U.S., is that right?
That sounds about right. Yeah, which is pretty remarkable. I mean, if I was looking back at the history of poliovirus, the deployment of the poliovirus vaccine, the sugar cubes and other, I think that in the U.S. took something on the order of 5 years to really eradicate, to really drive it down. Yeah. And then, with some updates of the vaccine along the way.
So in 1955, when the vaccine was deployed, there were about 30, 40,000 cases of paralytic disease a year, polio. And then by 1960, down to 2,000 cases, but still 2,000 cases a year. And people said, well, the inactivated vaccine is not going to do it. So they switched to the Sabin vaccine, the oral vaccine, and that got rid of polio. By 1979 was the last case of wild polio in the U.S., and that was it. So, but that inactivated vaccine, the clinical trial efficacy was only 50%, yet it was licensed immediately, and it did knock it from 30,000 to 2,000 cases. So I think a good amount of herd immunity there, too, kicking in.
Agreed. Yep. No, interesting, some of these parallels, and we'll see how things unfold here, but pretty remarkable to be at 50% adults after 4 months, 5 months here.
Now, Nels, what is the occasion for your getting a little barbershop thingy there?
The elephant in the room. So I've come out of my facial hair hibernation as part of moving forward here. Actually, the real reason here, Vincent, is you generously sent me a great webcam that I've deployed for the first time today. And I was, banking on that low resolution. I could kind of hide my facial features, but now it's, kind of high fidelity. So I'm trying to step forward here, and move forward. Kind of a spring haircut, I think, is the real reason behind this.
Looks good.
Thanks.
Good.
That's generous again of you to say. So should we jump right in, Vincent, to our paper?
Yeah, we have a cool paper today, don't we?
Yeah, we do. I'm excited about this. So the paper's title is An Evolutionary Portrait of the Progenitor SARS-CoV-2 and its dominant offshoots in the COVID-19 pandemic. And of course, we'll dive in here soon. But just want to point out the authors before we get going. So the list here, it's kind of a all-star cast, I would say, of computational evolutionists. We have Sudhir Kumar, Q-Quin Tao, Stephen Weaver, Maxwell Sanderford, Marcos Caboyo Ortiz, Sudeep Sharma, Sergey Pond, and Sayaka Miura are the two co-senior authors on this. And a lot of this group is headquartered in Philadelphia at Temple University, which has kind of grown into a hotbed, I would say, of computational evolutionary work. We'll talk a little bit about that. And then some of the collaborators here are at the Center for Excellence in Genome Medicine and Research. And this is at King Abdulaziz University in Saudi Arabia. So an international cast here working on this paper, reporting this. So, you know, the authors here are trying to take another attempt at sort of getting a handle on the diversity of SARS-2. And, you know, it's really amazing, actually, and unprecedented, I would say, is the collection of genomic sequences that are available to the public. So on GISAID, this kind of repository, was originally deployed for influenza and has been repurposed for SARS-2. If I'm remembering right, and I haven't looked in the last few days, but I think the total record is now, it's more than 1 million sequences. The analysis we'll be discussing today is a bit earlier, and they're doing some quality control. So the number of sequences that they're analyzing is in the hundreds of thousands, but still that's like, you know, for the amount of time here, it's a remarkable data set. And so there's been, of course, a lot of interest in what did the original SARS-2 virus look like? How is it diversifying as variants are emerging? What does this mean for the evolutionary process? Should we be worried about this? All of the above. And so the authors point out that, you know, some of the traditional techniques that you would use to compare sequences like this, so you're looking for signals of interesting positive selection or signals of adaptive evolution, that the virus, the mutations actually reflect improvements of the virus in its ability to infect, to spread. And so, but some of these current tools fall short, and for a number of reasons that they point out. So one is that it's sort of this compression of data, a massive data set, but over a short amount of time. And so low sequence divergence, actually, it's been less than 2 years since this came onto our radar and sort of shook up the world. And the average number of mutations is actually about 2 to 3 a month, which is relatively modest when you consider for kind of an average virus. There's some caveats to that kind of average, but the you know, almost a 30,000 base genome, and so not that many changes to track. And in addition to that, the authors point out there's, like, of those changes that you track, only a handful of them, or a subset of them, are actually useful for phylogenetics, meaning that they're actually sort of give you enough information to be able to infer the relationship between the viruses as they change. And that could be because the mutations are in repetitive sites, or a number of different reasons. There's also sequencing errors, right? So these are a lot of folks moving really quickly to try to get the data out there, which is great, and again, unprecedented. But then, you know, mistakes are made. There are actually some well-known now common errors that come up that are immediately filtered out, or sort of, you know, rejected by some of the screening processes of putting a new SARS-2 genome isolated from a patient into the database. And so the authors propose to take a different course here, or use a different strategy, a mutational order approach. And this is basically to try to infer the mutational history. So you also have timestamps here, right? We know roughly when we know exactly when the virus genome showed up in the database, roughly when it was collected. And so that gives us a time series, again, at really high resolution. And so what you can basically do is just, okay, which mutations were here at the earliest times and at what frequency? And then which mutations are just showing up yesterday and at what frequency, and then use that to propose an order of mutations, a mutational history, which mutations came first, and how did these mutations spread around the world. And this is not phylogenetics. So phylogenetics, right? You're taking a sample at the tips of the tree, the modern, sampling or collection of sequences from modern viruses, the ones that you collected today. And then you're using the comparisons of them to say who's most closely related to who, and then working backwards in those nodes to then infer something about the history. So really a complementary approach here.
And what I understand, Nels, is that if you want to figure out roughly when, the ancestral SARS-CoV-2 began to circulate, you can't use the tree because there's too much distance between SARS-CoV-2 and these related bat viruses, right?
Yeah, and so you're raising a really important point here, Vincent, which is the outgroup that you pick for phylogenetic analysis really matters, right? Because this is how you sort of, as you're inferring back to the early nodes in the tree, how does it relate to sequences outside of the all of the sampling that you're doing? And that turns out to be really important. If you pick different roots or pick roots that are not sort of as informative, it can actually change the branches that you infer throughout the tree. And so the authors, I think, rightfully point out that that's an issue here, an ongoing issue, is that in a lot of the sort of complementary phylogenetic analysis that has been done, sometimes folks have made choices on the rooting sequence. So some of the bat coronaviruses, which, by the way, the most closely related ones have something like 1,000 changes from the early recovered samples of SARS-2. And so if we're only talking about, a couple mutations a month, then that outgroup is way out there, so to speak. So why does this matter? Why do we care? So why are we spending this time, sort of kind of really dissecting a lot of these details? And so, it's really a question of what can evolutionary analysis teach us about the origins of SARS-2, and that'll be kind of, I think, the main conclusions that we'll get to after we step through some of the work here. What did the progenitor look like? Can you actually reconstruct or think about what an ancestral sequence looked like? Were the first cases, was it that ancestor? How close were we to the spillover of SARS-2 when it first emerged in terms of the quality, what the genome looked like then? So that's sort of that kind of sets it up a little bit. And it's interesting. What's really fun is that, so the mutational ordering approach, this isn't a new idea or a new technique. It's actually used pretty frequently, or it's been developed to teach us about the evolution of tumors. So somatic mutations happening in tumors, right? And so you can collect a tumor sample from a patient at one time point, and then the patient, will very likely undergo cancer therapy, immunotherapy, or chemotherapy, some combination of both, and then return to the clinic months or years later. And then you can sample, the tumor at another time point, and then sequence the genomes, and then use this kind of analysis of mutational order. So in the very earliest tumors, what was the mutational profile? And then the ones that are collected more recently, how has the tumor population, the cells in that tumor, that population, how are they changing? And are there sort of shared sets of mutations that emerge over time? And so the authors are borrowing or kind of repurposing from some of those tools to apply it for the first time to SARS-2. And so there are a few caveats here. And so one of them, which actually works really well, I think, for tumors, somatic cells, and might not work as well for SARS-2, is that you're assuming there's no recombination, right? So the idea here is really, you're wondering, are the tumors, are the sequences of the virus genomes, are they, as they're replicating and moving through time, is there sort of a vertical lineage here? Whereas they clonally diversify, they're not swapping genetic material. That can actually throw sand. Recombination, where you switch templates as the virus is replicating to another virus that might have a different set of mutations, could actually really kind of scramble the signal and make mutational ordering difficult. And so, they say that recombination is hopefully low, but that's an assumption. And actually, if you go back to our last live stream TWiEVO, number 67, corona chameleon, we talk about actually some evidence that even SARS-2, some of the B117 lineage, appears to be undergoing recombination. There are some signals of that. And so that's one caveat here. Despite that, they move forward and actually improve, using some statistical approaches to improve some of the mutational ordering tools by adding a bootstrapping procedure. And this basically allows you to be able to put confidence limits on the analysis that you're doing. So as you're inferring the mutational order, you can actually have sort of a statistical framework to think about how, based on the data, how likely is it that what you're proposing in terms of that mutational order, that mutational history, what's your confidence that you've got it right? And so bootstrapping, we've talked about this on TWiEVO before, not for viruses, but for other data sets. And you're basically just subsampling a smaller part of the data set and then asking whether the outcome that you're proposing still follows that same rules for each bootstrap, for each replicate that you do of a subset of the data. And if it all comes back the same, your bootstrap confidence is high. If it comes back different, then you're like, well, maybe our confidence is lower. Maybe we're not necessarily getting it right here. There's different outcomes that look equally plausible for, in this case, the mutational order, the mutational history. So anyway, they get set up, they're ready to go, and they do 2 runs of this. So the first one takes in the sort of probably a snapshot. It's also like sort of a scientific ordering scenario here where the paper takes a while to write and the analysis takes even longer to do. And so they were developing this last summer. And so the first set of data they hit from the database is from July 2020. At that time, I think there was something like 70,000 genomes, but they actually trim down. They do some filtering to a little bit fewer than 29,000 genomes. And this is because they want ones that are full genomes with some quality to them in order to have some confidence here of what they're looking at. And so that first analysis, and then the second one, kind of on the heels of that, after they test drove their MOA, mutational ordering or mutation order accumulation analysis, they did almost 70,000 genomes. This was last October. And it's kind of interesting. So anyway, using these 2 data sets, they get what they call mutational fingerprints. So they propose roughly 50 mutations of high enough frequency and that move through, or they're found in the genomes as the virus has moved through the world, as it's spread through the world. And these are sort of 50 informative mutations. There's a great table that just says, which gene is it in. It's not always a spike. Other parts of the virus genome, what the mutation is. And then here's where, we've talked about this before we get into the alphabet soup, Vincent, and it gets even worse here. So we've
they have Greek soup here.
That's right. This is Greek alphabet soup. So they have this 50 mutations. And as they start to put together these fingerprints, almost these barcodes of the combinations of mutations as they show up together in the virus genomes, they classify them with Greek symbols. Mm-hmm. And so, there are some echoes of some of the nomenclature we've already been using, B1.1.7, some of the lineage stuff. There's a little bit of overlap in some of, obviously, of the mutations and the way that we define these sort of clouds of variation or lineages of the virus. But so they've got a whole new Greek alphabet for if you really want to go into the weeds. So if you look at figure 2, and I believe, so this paper is now kind of in preprint, or it's more than a preprint. So it's been peer reviewed. It's accepted at the Journal of Molecular Biology and Evolution. And the PDF is available at their website. I think that's open access. I hope it is, but if we'll put it on the link after the show, if you really want to dive in. So if we look at figure 2, and I don't know, maybe you can even pull this up, Vincent. Fantastic. So this is, the mutational history graph that they, after doing those 2 runs through of the data using the MOA approach, and they have so it doesn't look like a phylogenetic tree. Here we go. Yeah, this is great. So it looks like these circles with arrows between them, and so working from top to bottom, the mu1. This is the very earliest of viruses collected. Yeah. And so these are some of the, also called the Wuhan 1 collection of viruses that were deposited early. And there's this progression. So and these sort of offshoots from the arrows, which are the differences in mutational fingerprints that they see, the mutations that they actually see as they look through the genomes. There's a color code here based on where in the world that virus genome was collected from. Red is China, blue is North America, for example. Green is Europe. And then you see, so all the color, so there's that color coding, and then there's also the amount of the circle that's colored is the frequency of the mutation. So as you now, if you look at the top of the graph, these are fully colored circles, whereas if you look at the bottom, you can see ones where like three quarters of the circle is not filled in, and that means that's exactly what you'd predict as you get to newer or more recently collected genomes. These ones, the mutation hasn't had that amount of time to get through the whole population. Whereas for the very early ones collected that were the sort of the very early ones that then spread that were successful, those mutations persist. And so basically all of the genomes have that change, have that substitution from the reference that was collected. And I believe the reference here that they're using, I have to double check, but is Wuhan 1. Mm-hmm. Yep. So that gives you, and then, you can kind of stare at these graphs and think about what does this mean? It's not always totally intuitive. It's a little bit of a different way of visualizing the changes over time, but that gives you sort of a roadmap of what this begins to look like. And then, now that they have this all together, they could actually start looking at the types of mutations that are accumulating at high frequency. And so one of the common sort of pastimes of evolutionary geneticists is to determine of those mutations, do they actually change these are genes, right? Coding for proteins, and are there amino acid substitutions? So when you make that base change in the RNA, does it change the amino acid that it codes for, a non-synonymous change? And the ratio of that versus the silent changes, where you make a mutation but the amino acid is the same, because the genetic code is degenerate. You can code for the same amino acid with more than one 3-letter codon. And the number they recovered from their data sets, the snapshots of the data they took at different times, was 1.9, a ratio of changes non-synonymous that change the amino acids to synonymous. That's high. And actually, if you do a sort of traditional analysis with one of the tools that a lot of the authors had developed, this package called Hi-Fi, it's on their website, datamonkey.org. And that's actually, you can find this, I think, tool there as well. Mm-hmm. They note that if you just take some of those closely related bat coronaviruses and a collection of those, the ratio is much lower. And so if you dig in, it gets a little more complicated. It's hard to say too much about that, but not totally inconsistent. So when you have high ratios like this, the virus is coding for different proteins. It's changing rapidly. It can be a signal that the virus is adapting. Probably, I think, more impactful from the analysis they do, the mutation accumulation, is their ability to actually work backwards. So they start at Wuhan 1, and then they work forward to try to order the set of mutations. But then you can also, once you have that data set, the timestamps, you can also work backwards. And so what they note is actually the first, and other groups have noted this too, is that the first viruses collected aren't the index case, in the sense that this isn't the person that where the spillover event happened, or the virus moved from one species to human, and that comes out of their analysis, comes out of other analysis where there are already differences in the index cases. 3 mutations, I believe, out of their 50 that are different, even among the very early ones that they collect, and so they can work backwards to infer what they call the pro-CoV2, or progenitor of CoV2. This is also the nomenclature you get here is oftentimes the MRCA, or most recent common ancestor. And so that also, because they're doing those timestamps with the sampling that's happened ever since the index cases forwards, you can also do that backwards. And so when they do that, they infer the emergence of the virus actually 6 to 8 weeks before the index case. And that would put it at roughly October 2019. Other groups have said this. It's probably almost just a good sign that their methods are, kind of bringing back a consistent result there.
Now, what limits how far back you can go?
Well, so you can go back as far. Well, so you're in this case, you're going back to this sort of idea, almost this philosophical idea of on the day the virus spilled over, what did the genome look like?
Yeah.
Um,
Yeah.
you can go back, but of course, that's sort of an arbitrary um, choice to make, and we're interested in it because it involves our species and obviously the start of a global pandemic. But of course, coronaviruses, that lineage goes back farther and farther, and so you could start to go back. Can you get closer to some of the bat coronaviruses, like 1,000 mutations back, and you could add that virus in your. But what happens is your confidence goes out the
Yeah.
window,
Yeah. We
right?
don't have a lot of sampling. That's the problem, right?
Exactly right. And this is why, for reasons other than just political ones, that we'd like to know, where did SARS-2 come from originally?
Yeah.
More sampling also, that's something that evolutionists are excited about or have some interest in, because it allows you to make more more high confident inferences about sort of the evolutionary history of the viruses, not just back 6 weeks, but back 5 years, 10 years, something like
But
that.
you would need, if you wanted to go back to the bat ancestor, you would need to have a lot of bat viruses sampled. I mean, we don't have anywhere near the same number as we do for these human viruses, and that's the problem right now.
Correct. Agreed. There's a massive gap in the phylogenetics between the two. Exactly right.
Yeah.
Yep. So
and it's not even clear where you would go to sample those, because they're finding SARS-CoV-2 like viruses in other countries now, in Thailand, in Japan. Yeah. And so it's not even clear where you would look, right? And that makes it really difficult.
I agree completely. Exactly right. It's difficult. So space is vast here. Time is a little bit vast. We're probably talking in the 5 decades of virus diversification. And so that's a big challenge. It's one I think that's obviously worth pursuing, and hopefully there is continuing to be pursued, and hopefully an increasing resources are put to that. But yeah, so so why does it matter so much that we're inferring something more high confidence back to October of 2019? And I think for a couple of reasons. So one is actually, it allows them, and as they're looking at their mutational profiles, their kind of key mutational fingerprints, as they call it. This allows you to actually see infer that SARS-2 is moving kind of under our noses back around the turn of the year from 2019 to 2020, and to see that there are a couple of cases in the U.S., for example, in Washington State, in New York, a couple of cases in other locations in China that already had these early sets of mutations. And so, it was spreading before we were onto it. And of course, that kind of recent history is also being put together too, as as people were scrambling with this unknown disease, these pneumonia-like symptoms that turned out to be SARS-2. The other really, I think, important, probably even more important outcome, at least from an evolutionary kind of genetics standpoint, evolutionary virology standpoint, is that now that they can infer what the progenitor sequence looked like, not just when it kind of arose, but now you can use that to root your trees. And that's certainly a massive improvement over picking other bat coronaviruses that have thousands of changes. And so then as you kind of return to phylogenetic analysis, and as more evolution is happening, as the viruses continue to diversify, you can actually do better in your phylogenetics by using that progenitor sequence. There's a better rationale for using that as a more closely related outgroup. So then, if we go to figure 5, this is a look at their mutational. Oh, actually, yeah. Oh, before that, actually, Vincent, if you don't mind scrolling to figure 4.
Sure.
So here's another one of these plots, and now they're looking at kind of the backbone of mutational history. So, kind of more of a 30,000 foot view. And this just shows, again, sort of the geography of this as the virus is spreading around the world. And this is A and B. This is their 2 data sets. So the first one that involved, about 20,000 sequences, and then the second one that involved more like 70,000 sequences. And what you can see, very plainly, is that there's more circles on there, and that makes sense, right? The longer the virus goes, the more mutations show up, and those are out at the sort of bottom of the progression here. And those arrows are, they say something about the link of where how these viruses are moving actually, or how they're related to each other and moving both through time and geography.
One thing that's worth pointing out, Nels, is
Yeah.
that the sampling in Wuhan, the very early sampling, revealed already 2 lineages, and that's what they're building on here. And so this makes sense, right? That what Wuhan 1 is not near the beginning. It's somewhere downstream, as you said, maybe 6 weeks. And so it's not surprising that there was more than one lineage there, right?
Exactly. And then that blue dot that you see to the far left in both A and B, those are the very first cases sequenced in Washington. And so, there is, and I think in the epidemiological sense, we don't know the index case, and inferring that these ones were index cases, we kind of were a little bit down the river on the virus already starting to circulate. So then let's go to figure 5, where you were there just a second ago. And this is showing the mutational fingerprints. And so this might look a little bit familiar. So we've spent some time looking at some of the websites like covariants.org, and there you're looking at, so now we're looking at the percentage of genomes that's working from bottom to top, color coded. And of course they're using their mutational fingerprints. So they're 50 or so Greek letters and combinations of the Greek letters as the mutations are on the same, on the same genome. And then on the right is a sample count. So, and that's important. That's sometimes I think we under report that. How many, when we look at the proportion of the viruses circulating, how they're related to each other, what's the sampling that's sort of behind that? Because if you're at low sample numbers, some of the patterns could be really noisy. You might see something and say, oh my goodness, this virus, this variant is dominating. And yet if it's based on very few sequences, it might just be more of sort of an ascertainment bias. And so the 3 graphs here, the top one is the virus types or the fingerprints in Asia. The middle is Europe and the bottom is North America. And so you already can probably see some patterns that look a little bit familiar. So in the middle panel B, kind of already, this is sort of September, November 2020, moving into the early days of 2021 in that sort of brown shaded area. This is B1.1.7, that lineage starting to dominate Europe. And in North America, the authors actually point out, this is a little bit, a little bit historical already because we're moving so quickly. But while B117 was spreading in Europe, the US, that blue area was staying pretty constant. And this is by mutational fingerprints. Mutations had already accumulated, and the virus was just, with that set of mutations, with nothing else, was doing quite well for a long amount of time. And I think this starts to maybe give a little more context. We often, when we're talking about, in the kind of ripped from the headlines and the new variant that's arising currently, of course, a lot of interest in the one B.1.627.2, which is a lineage that recently started spreading in India and beyond. But I think this kind of analysis starts to actually, so that collection of mutations, it's not just strictly, it ends up overlapping very much with the definition of a lineage, but it also has it sort of in the context of all of the mutations that are accumulating over time, or the mutational order. And that gives, a little bit of context to how this is stepping through time as a progenitor. Kind of, we almost have these variants drop. It almost feels like they're dropping out of space, this new super mutation or this new, and kind of almost these breathless headlines. And I think it's important to remember that this is all the same virus. This is all SARS-2 with sets of mutations that for many reasons could be sort of gaining in numbers in different locations over time.
And so now's this, I'm not clear why North America doesn't show an invasion of the brown variant there.
Yeah, so that would, if you fast forwarded, exactly. And so there's a threshold here for their mutational profile. I think it's like 3%. And so probably just about that time where and then the way it's defined, it's not, so this isn't strictly like they're a one-to-one match of the way they're classifying the mutational footprints. That doesn't overlay perfectly with the variants themselves. And so
Okay.
some of those mutations from B117, for example, which is now, I don't remember the latest, it's something like more than half, certainly 60% or even higher now in the US of cases, but that are getting sequenced. But yeah, so that's actually captured in there, but it's just represented differently, and it's at a low frequency as well. Yeah.
So a couple of people are talking about, finding patient zero, and obviously that's of interest. The problem is that, many of these infections are asymptomatic, and so it may be very hard to do. We talked on TWiV today with 3 members of the WHO committee who are investigating the origins of SARS-CoV-2. And they said they have a lot more work to do. For example, they're going to be looking at banked blood, which is going to be provided by blood banks in China. And they can go back a year to see if they see antibodies to this virus. And that may help provide, additional information. So it's not easy, but and I think the other issue here that's quite interesting is that there's an early isolate from Italy, which, a lot of people didn't quite believe. And now it looks like, I think that's from December. And if this prediction is correct, they're saying that the virus was already circulating in Europe and North America in December. So that may be right. And unfortunately, that piece of RNA from Italy didn't have any of these signature mutations that they could use to verify, right?
That's right. Yeah. Yep. No, that's exactly right. And I think so. In addition to, the WHO investigation, this also points to the value, as we were already discussing, more sampling as well in potential animal reservoirs, and seeing if we can start to close that gap between the viruses that we are, the most closely related viruses to SARS-2 and that sort of 1,000 mutation gap that currently exists with the closest known ones.
I mean, we have to point out that the sampling is extremely inadequate. This lineage, this SARS-CoV-2 lineage, has been circulating in bats for over a decade, unnoticed by us. We never sampled it. And, they do some sampling in China and other countries, but obviously it's nowhere near enough to pick this up. So it just points out the deficiencies, and we need to fix that.
Yeah, it's really interesting because it's all of these sort of like unbalanced situations. So we have, on the one hand, I 100% agree. We're really undersampled on coronavirus diversity in general, but then at the same time, there's almost this like wealth of sequences that we've accumulated this in GISAID over the last year, which we've never done before.
Never.
And I think, and that's, and that's also caused a little bit of, I think, some issues with perspective on how do we, there's the old saying, the old cliche, if you're a hammer, everything you see looks like a nail. And so if you have all of this data, it starts to, we start to drive a lot of nails, potentially, not all of them necessarily pointing us to sort of real biological meaning or real biological impact. But so that covers this one. I think, and it also, I think one of the reasons I picked this paper and wanted us to spend a little time chewing on it was to also just point out, so first of all, how quickly everything is moving, as we've talked about before. We're kind of looking at science in real time, but then how even when you're moving fast, I think science takes some time, to build a new tool, to be able to, or to repurpose a tool to then use it for this kind of data. Here we are kind of a year on. And so this also illustrates how I think sometimes how science moves at a pace a little bit slower than certainly like the news headlines. And in terms of the data that we're continuing to generate as we're, almost doing this natural experiment with deploying the vaccine at different sort of tempos in different geographical locations, some areas going really fast, other areas still not out of the starting gates in many cases around the globe. We'll be looking at this for generations to come of science ahead in learning, I think for decades to come. And so this sort of this slower pace of building more careful tools instead of just slapping in a root, to actually sort of spend some time, some months to get a high quality progenitor sequence inferred and then to continue to build on that tool. The nice thing about this tool is it's useful not only, of course, for this, but as the data sets continue to grow, and then also, of course, for other, especially as we, genomic epidemiology advances, not just for SARS-2, but for other viruses, potentially in pandemics ahead.
Well, I think the point is well made that this analysis was done because of the shortcomings of all these data that we have from the human isolate sequences, and now going forward, this should be used in other outbreaks and other viral analyses, because as you said, it was used mainly for tumor genome analysis. So this is good. It's an outcome of people saying, "Hey, this phylogenetic tree isn't right. Let's fix it." So I think that's great. I think
yeah, absolutely.
great.
Yeah, and it also shows, some of the existing tools. Like it's easy. So again, for speed, you'll take an existing tool and use it. But for understanding, that's sort of a different. That's really a different pace. And I think sometimes sometimes gets a little bit lost in our conversations. So, Vincent, speaking of conversations, a little bit of controversy as we sort of step through new variants. What we've called from time to time variant fever. Ideas about are the variants of concern, are they increasing in transmission? Are they increasing in fitness? And I wanted to ask you if maybe we could step through that a little bit, what you're seeing in the conversations on TWiV and in other places as you're both following and reporting on the pandemic as it unfolds.
Let me do that, but let me first acknowledge a couple of contributions from Rob Super Chat. Thank you, Rob. It supports our expenses here on TWiEVO and all of the Microbe TV podcasts, and also JL. Thank you for your Super Chat. JL's from Paris. Really appreciate it. I heard somewhere someone said, if you produce something and people really like it, they'll support you financially because it makes them feel good. And, we need this support for our expenses, but if you would like to feel good to support us in general because you like what we do, please go ahead. You could either do a super chat here or microbe.tv/contribute, or you could just give us a thumbs up here on the live stream. That helps too. All right. So as far as variants go, so I have a couple of perspectives on this. First of all, the mainstream media has been going crazy. Every variant is more transmissible, more virulent. The end is in sight. The vaccines are not going to work. I think this narrative is really unproductive. And I wrote, so there's a writer over at the Times, Apurva Mandavilli.
Yep.
And I wrote and I said, your narrative on variants is wrong. You're calling them all more transmissible, but there's very little data to support that. And she said, well, you're the only one that feels that way. And I said, no, that's not true. So I sent her a couple of names, including Nels, and then she interviewed him. I didn't want to talk to her because I was too not angry, but just disappointed. But anyway, my feeling has always been these are more fit variants, and therefore that's why they overtake ancestors. Yeah, some people call them scariants. Yeah. And one of the people who agrees with us is Ron Fouchier, who is at the Erasmus Medical Center. He works on transmission of influenza viruses. So this is a guy who works on transmission. And he says, we see this all the time with influenza viruses. We see variants arising, and they have a very slight fitness advantage, probably because they get around neutralizing antibodies slightly. And that's enough to drive them through the population. But we never say they're more transmissible. And I think that's really the lesson here is that they're not necessarily transmissible. I mean, they could be, but no one's done the experiment to prove increased transmissibility. And I'll tell you what people do. And for some of these variants, for example, you will hear people say 100% more transmissible. And I just cannot deal with that. And to have more transmissible variants arising all over the world, it's just doesn't make sense. So what they do is they measure RNA shedding in nasopharyngeal swabs, which of course is not infectious virus. So it really doesn't inform you about how much virus is being shed. And then they look at the increase in the number of cases, and they use that to modify the reproductive index, the R naught, However, that's not correct because R0 has a human component. So if people start to go to bars and restaurants, that will cause an increase in transmission, but they never factor that in. So I would like them to do other experiments to prove increased transmissibility and not simply fitness. Ron Fouché said fitness is not the same as transmissibility. They're distinct. So I'm trying to diffuse this narrative, okay, and get people thinking about fitness. And I'm seeing a little bit of it here and there, but I'm on a number of phone calls every week, and people are still talking about increased fitness. And you know what it is? The original report out of the UK said increased fitness, and then people just repeat it. They hear it, and even Tony Fauci just repeats it. He doesn't actually look at the data and see if an experiment has been done to prove it, but just kind of repeats it. And I think you need to be a little, what is it that I tell people? Just be a little more skeptical, and the virulence increase has been disproven for the UK variant. I mean, a Lancet paper came out saying, no, there's no increased virulence. Another preprint is out saying the course of the pandemic in the UK was not altered by the variant. So, I think as we do the right experiments, we're going to get the right answers with time. My feeling is the vaccines work against the variants. That's, we're seeing in the U.S. massive decline in numbers. And I think that's the bottom line. The variants affect B-cell epitopes, but they don't affect T-cell epitopes. And that's what prevents you from serious disease and death and hospitalization. And I'm sorry, Nels, to go on, but you kind of asked for it, I suppose.
I love it. I did. I asked the question. And no, I think so I've been thinking about this a little bit too. And, I wonder, and this gets back to kind of, one of the reasons you and I started TWiEVO together is how do we discuss sort of ideas or even define terms between different fields. And so fitness for evolutionary biologists can be kind of a loaded term as well. But transmission is it kind of strikes me that transmission is kind of a broad and probably imprecise term, or it's defined differently, maybe is a better way of saying it, by different people. And it encompasses so much, right? So I did it right before the show. I did a quick, Google search. And so I'm not going to stand behind this to my death or something, but a definition from a paper on virus transmission. So a virus must enter a host through a portal of entry, replicate or disseminate within the host, and then be transmitted to a new host through a portal of exit. There's a lot of there's a lot happening in that sequence, right? So there's a lot of happening. First, the exposure event from one creature to another, or from maybe an environmental sample, whatever. However, you come upon a virus, and then it getting into the onto the surface of some tissues, ultimately into cells where it's replicating, getting out of the cells, disseminating into, and then the transmission event to another creature, human or otherwise. And in addition to the biology there, I think as you've pointed out, there's a lot of behavior in there as well. And so, when we talk about, I guess maybe to me, it really matters what part of that transmission cycle is being altered. And I think maybe some of the, and that's really hard to know, right? And again, takes time. So in the news headlines, I don't think are maybe the most sort of amenable to that nuance or to that, that understanding, right? Yeah. And so
the transmission is a poorly defined term, right? Because, as you said, there are a lot of steps involved there. Someone asked, how do you prove increased transmissibility? I think the question is, how do you prove increased fitness? And how do you say what aspect of this cycle that Nels just elucidated, what aspect is improved in the variants? And I have to say, you're not going to find an answer overnight. It's going to take some time. You're going to have to do experiments in the lab, and you're going to have to do animal experiments. And in the end, it's going to be unsatisfying because you never know if it applies to what's going on in people. So even with influenza, Fouché said, we think it's antibody evasion that's giving slight fitness advantage, but we can't prove it because what we see in animals is not necessarily what's happening in people. And I think part of the problem, Nels, is that the this is a pandemic. It's an emergency. So the press is freaking out. And so they want to grab anything, and they don't understand the nuance, and they don't understand you got to wait until we get the right experiment. You cannot jump on the first report, and you cannot speak to 10 people, and then if 8 say something, you go with that. That's not how science works. You have to present both sides of any particular picture.
Yeah, it gets complicated. That's for sure. I wonder, and part of it. So, I mean, the current news headlines, of course, are, the variant B1.627.2. I think I got that right. That's really on the rise in India, and then again, of course, there's all kind. One of the moving to the UK, where there's all kinds of great surveillance, relatively speaking. So basically, a pretty high proportion of patients will have samples taken in the virus actually sequence, and then seeing a bit of an uptick there of that variant. Here, I think one of the caveats that sort of doesn't get discussed a lot certainly it's not found in the news headline, right, is that what are the numbers here? So in the UK, this is, the bigger picture is this actually massive success story where numbers of people infected is way down. And so here then, the amount of sampling, which is relatively good, it's still a small sample size.
Yeah.
And so the confidence interval on some of those observations is actually, I think that's kind of lost in the conversation as sort of the energy is put into, wait a minute, we should be really scared with what's going on here. So that your point on fitness as well, I think relates to this. I mean, so one of the quick assays that scientists can do is how effective are neutralizing antibodies to a variant of interest or a variant of concern. And so, the good news for most of the vaccines is that the neutralizing antibodies that they stimulate are still quite good against every variant we've observed so far. And because the vaccines were so successful at the outset, better than I think some vaccinologists dreamed of, there's still a good efficacy window here where we're doing quite well. But yeah, I think the fitness point, or as the viruses are competing, it's harder to be a coronavirus. I think we've said this on TWiEVO episodes before, because of both natural natural immunity and supercharged immunity from vaccines. The human population has also wised up. Our behaviors are a lot different a year ago than they are today. And so, yeah, that's actually another reason I think, getting at the progenitor virus, or remembering the genomes of the very early ones compared to some of the mutations that have accumulated, and actually setting up experiments to compare them almost head to head. That starts to get at how these viruses in the wild are competing with each other against the potential hosts, which have I think in an unprecedented way become much harder to be hosts for this virus. And that's some of the success stories we're seeing in Israel and other places where the vaccine has been deployed.
I think part of the issue, Nels, is also that this is a BSL-3 virus, and not everyone has a BSL-3 lab. So, fewer experiments can be done as a consequence. And I think at some point in the future, this will be probably a BSL-2 virus once everyone is immune and there's much less disease. And then maybe we can get some of these answers. But there's a comment here, vaccine breakthroughs is part of the variant scare. Yeah, it is, because breakthrough happens with every vaccine. And breakthrough is a horrible name coined by I don't know who. But every immunity, whether it's naturally acquired or vaccine acquired, you have infections, and we don't call them breakthrough. The polio vaccine, as we said earlier, was only 50% effective. Nevertheless, within 5-6 years, the number of cases were way down. And so what happens with a vaccine, a year after you get it, you have very low antibody and T cell levels, very low. Waning is normal, folks. Now they're making a big deal out of this in the press, but you know what? It's normal. And then when you get infected, the virus is going to reproduce for a couple of days until the memory response kicks in. And in those days, you will have a little shedding, but probably not enough to transmit. and you will not have any disease, or you're going to have a mild disease. And so breakthrough is wrong. It implies that there's something wrong with the vaccine. That's the way immunity works. It's not, for the most part, sterilizing. Sorry about that again.
No, no. It's like you've been in some other conversations almost. Like you have a, no.
I want to thank Sandy here for her very generous super chat. Appreciate it, Sandy.
Wow.
Nice
Yeah.
endorsement.
Thank you, Sandy. I really appreciate that. Yeah, I lost my train of thought actually. So,
Sorry.
no, not at all. Not at all. We were talking about, so the notion of breakthrough, right? And yeah, that's a more nuanced thing as well, right? Because, and I think you pointed this out before, but so because there's so much high asymptomatic spread of this virus, and then that's probably even like that's, we're pushing that even farther into more asymptomatic spread, given vaccine protection. And so I think, the CDC has decided recently, just in the last couple of days, to stop tracking asymptomatic spread among vaccinated patients, because it's sort of, you're looking at something that's just becoming so common and sort of under the radar of clinical issues, that's already starting to fall off. And then, again, those are technically breakthroughs, right? And it sounds much more dramatic, even though it's something that you might not even have a cough or a sniffle associated with it. So yeah, I think it is. I think things are a little, so in that, that article you talked about from Apurva Mandavili, and it was co-written by Carl Zimmer. So I spoke to Carl, and yeah, just what's driving the news headlines, and have we lost a little perspective with some of the discussion about variants and transmission and other of these terms as we're moving. It's important. It's just the speed of science and the speed of the news cycle, I think, are well out of balance.
Did Zimmer get it?
Yeah, so it's an interesting article. So this is from a few weeks back in the New York Times, sort of 4 months on from late December, when B.1.1.7 was first coming on the radar, and what happened. So there were some pretty strong predictions that the U.S. this would be kind of what happened in Europe, and that didn't happen. I think we were underestimating the competitive nature of the existing variants here in the U.S. or others that sort of arose on that same time scale, some convergent evolution happening as well. And so I thought it was interesting, some interesting quotes. And I think actually the best quote in there, I thought, was from a colleague, Sarah Kobe, who's at the University of Chicago. And her quote was, we don't know a lot about what we're looking at here. We're learning as we go. And I thought that's sort of lost, I think, sometimes in our conversations too, is the idea that we could admit that we don't know, as scientists, that we don't have all the details. We're learning as we go and adjusting to evidence as we see it in real time. And I think that also might be within slightly mismatched with sort of the news cycle, and the notion of certainly the politics, where definitive statements are kind of the bread and butter of some of those conversations. And science, I don't think, is always equipped to sort of work well in that space. If you do really go out on a limb or make a strong statement as we're figuring out stuff on the fly, which is just how science works, then there's going to be mismatches in the discussions that we have. I think that's leading to a lot of issues currently, honestly, not just as we think about variants as the pandemic continues, but also as political conversations unfold around the notion of where did SARS-2 come from, and related questions like that. Getting to your point, Vincent, too, and worrying about sort of breakthrough is, I think, some great already, a side effect of these RNA vaccines is that they're programmable, in the sense that we know that, like, as we're analyzing these mutations as they accumulate in order, we can put those into, especially like in the spike protein, we can put those into modify the codes that are used in the messenger RNA vaccines, whether it's Pfizer, Moderna, and actually think about boosters, which is another sort of well-worn path for vaccine history, for vaccinology, is booster shots, which we're all familiar with as well. And of course, that'll probably be in the cards here for SARS-2 as well.
Someone asks, why are many virologists supporting variants behind increased cases and transmission? Because they're just repeating what they hear. They're not thinking independently. Someone said it's increased transmission, and so they repeat it. But if you go look at the data for it, as I said, it's not very good. And that's the bottom line. And yeah, I'm an outlier. That's what drives science in some cases. So is Richard Feynman an outlier. I'm happy to be in the same category.
Well, and as the virus spreads, it has to be some sort of variant. There's no choice with that. And again, yeah, I agree. I think in that case, small numbers, right? Sampling is pretty modest in a lot of these locations. And so some patience to see how this will unfold. Yeah.
I think you need to be somewhat provocative and get people to think differently, and then that changes the paradigm. When everyone is following the same narrative, it's never good. But let me just say that I've been thinking about this for 40 years, so I'm not always right, but I have some perspective, I think, and I think a lot of people don't have that perspective. And part of the issue is also that a lot of people are new to virology because of this pandemic, right? Yeah. And we have a lot of people coming in and not really having a history of thinking much about viruses and how they're behaving and so forth. So I think that's part of the problem. And so I'm just sticking to what I've been trained to think. That's it.
No, I hear you. And actually your point that you made a few minutes back, a really good one, which is just so that if we look at the field of virology, the number of kind of quote unquote card carrying coronavirologists before this pandemic was really small. It's kind of ballooned in ways, but you can't just sort of immediately transplant that experience, that history. It takes years to learn to get to know a virus, how it operates, how it replicates, its tricks, its genome, all of these things. And so, I think that's changing pretty rapidly, but that takes time as well to have a base of expertise in order to really, again, bridge that gap between, what's happening in an ongoing pandemic versus our understanding of the virus itself, how it evolves, how it operates, how it transmits. So anyway, I really appreciate this part of I really appreciate this part of the discussion. I think it's important, and kind of why we do TWiEVO is to try to work between the cracks here, between the different approaches, between the different fields, and hopefully move the conversation forward. And so speaking of that, maybe we should move forward to the mailbag.
Yeah, that's a good idea. But I can't help but saying, someone said Please. I'm being dogmatic, but that's not true, is it, Nels? I'm not being dogmatic. I'm just looking at the data differently. And if the data change, I will change as well. That's not dogmatic.
Yeah, and it's also, I agree. I think it's also, we kind of get drawn into the conversations that are happening, right? And if prevailing opinion or ideas are coming in kind of hot and fast and strong, it's like, I think it's a very common response for us to try to meet that the information that's coming in with sort of a similar argument, even if it's on the other side, a similar. And so, dogma can be dangerous for any conversation on all sides. And so I agree. I think that's not what we're doing here. I think we're hoping to point out what some of the nuances where we need to focus some energy to learn more. And it, just as you're saying, Vincent, having that adaptability as new information comes in, it's not a weakness to change your view. That's kind of defines the scientific approach.
Now, before we go to the mailbag, let's address this. How should we react to the lab leak theory? So you should not believe it. There's no evidence for a lab leak. And today on TWiV, we had 3 people from the WHO committee talk about this, and they provide some very compelling evidence. They said, look, if you have some evidence, scientific evidence about a lab leak, please give it to us. But we haven't seen any. And next, the Friday, we're going to have Robert Gary on, who's written a lot about the lab leak hypothesis. So I think you should disregard it. And
This will be on TWiV next week.
Yeah, so this 2 TWiVs this week. They'll both be released. One is released at midnight tonight, and one will be released on Sunday. So listen to that. But I think the lab leak hypotheses there's no evidence for them. Okay, there's zero evidence. It's not impossible, but there's no evidence for it. And all the Nicholas Wade, for example, doesn't know what he's talking about. He says the furin cleavage site is a red herring. No, it's not. And in fact, that's my pick today, as you will see.
Oh,
But,
cool. Yeah, no, I'll weigh in a tiny bit on that too. So, and since our last live stream, there was this letter that got a lot of notoriety in the press from a number of notable scientists: Ralph Barrick, Jesse Bloom, a number of others, Christian Anderson, et cetera. Actually, no, Christian Anderson I don't think was on that, but it was weighing, sort of holding open that conversation with limited evidence, sort of across the board. Really calling for ongoing, just as Vincent is saying, calling for ongoing investigation to try to get to some evidence, or to at least have a better sense of what's out there. Really thorny. We'll return to this too in my pick of the week as well. But yeah, there is a little bit of on thin evidence, but sort of heated topics. I think things can run off the rails pretty quickly.
Yep.
All right, so let's move to the mailbag. The first email is from Kaim, and he writes, "Hi, Professor Eldy. As far as B-cell evolution goes," this is from a conversation we had a couple TWiEVOs back, "I'm with Dave. B-cells are indeed the relevant organism, and the fact that you don't pass your evolved B-cells to your children is irrelevant, in the sense that the B-cell does in fact have its own germline. A unique VDJ rearrangement with somatic hypermutations is passed down to its daughter cells, but cannot be transferred to an unrelated B cell, by which I mean one descended from a different naive B cell with its own unique rearrangement. Moreover, while it is of course precisely this germline of which most selection acts, changes can accumulate in other portions of the genome that can also increase reproductive fitness, otherwise known as a lymphoma. Cancers evolve too, at least by this way of seeing things. Whatever we call it, though, thank you for an always insightful and interesting podcast. I look forward to it every month. Best, Kaim, who's a computational immunologist specializing in B-cell evolution." So yeah, thank you for this really nice note. And it is, I think I agree with you. To take a little bit of a bigger picture here. And in fact, the paper that Vincent and I were just discussing, uses this mutational ordering from cancer evolution and kind of repurposes it to viruses. And so I think it's absolutely true that, using the concepts of evolution, of course, or mutation accumulation, agree 100% that within the genome of a B cell, in addition to sort of a useful VDJ rearrangement that will then be passed down to daughter cells, that you could have other sort of fitness effects at a somatic level, for sure. And so putting that under the umbrella of evolution, I'm totally with you. I guess I was probably drawing a distinction between sort of this long running evolution that would go beyond the length of the lifetime of the host of those B cells. And so I would say both are evolution, the evolution that runs for generation to generation and beyond, longer courses of evolution, but still very much possible to borrow ideas and from these different processes and learn about the biology, I think, in very meaningful ways, which is exactly what's happening here. Actually, I'm a little bit curious. So I think the rule stands, which is that the B cells are unique. They're not going to pass their genomes or genes from one B cell to another. But I wonder if that rule is ever broken, if there's somehow horizontal transfer between B cells. That would be a fun topic. Super rare, I'm guessing, and hard to document, but that could be pretty cool.
There are some B cells. There are some immunoglobulins that are in the germline, right? So if a pathogen puts selective pressure on an organism, then the relevant rearranged antibody genes will be incorporated. And we can see that for some pathogens. For example, respiratory syncytial virus broadly neutralizing antibodies are encoded in the germline of humans, but not HIV antibodies. So my interpretation is that RS virus has been around for a long time, has put pressure to select for humans that have integrated those DNAs. I don't know if that's the right theory, but it seems to make a lot of sense. So I think under some pressure, you could incorporate those into your germline.
Oh yeah, no question. So this is just distinguishing the somatic hypermutation, but of
Yeah.
course in our germ, yeah, no. So the whole system, right, that's encoded in there, that reflects this longer running evolution. And yeah, I agree. Changes to it, or sort of even at the baseline level, it might be tuned for
Yeah.
more
Yeah.
viruses than others, and depending on the species. Yeah, absolutely. Yeah, it's a good point.
Well, it makes sense, right, that it would do that. Let's see. The next one's from DB Nelson Vincent. I just received my second dose of Pfizer. For about the first 12 hours, I felt a little buzzy or giddy. You're probably really happy about it.
Yeah.
Several associates reported feeling similarly. It brought me back to the rats and the analgesia papers that were previously discussed. And DB provides a link to this paper that was published in the journal Pain. SARS-CoV-2 spike protein co-opts VEGF-A neuropilin-1 receptor signaling to induce analgesia, right? To, deaden pain.
Yeah.
And so back to DB. I'm wildly speculating here, and I have no idea if the spike produced by the vaccine could get into the bloodstream, but what a story if somehow early in infection the virus actually made you feel good. Maybe you want to go out, maybe go to the bar with your friends, and maybe you're a little more talkative than normal. Meanwhile, you have pre-symptomatic transmission going on. Sure would be a good outcome for the virus. Yeah, well, I think that's a human view. That is a human view of the sequence of events. It is not necessarily what actually happens. The Pain paper, it's not clear if this happens in infection. It's, an in vitro experiment. I can't remember where we discussed this on TWiN or TWiV or whatever.
Yeah.
But we concluded, maybe here on TWiEVO, I don't remember, but we concluded that there wasn't enough evidence to really support that. And, viruses changing behavior, it's a popular topic, right? Because it sounds really, really good. But I think if you are asymptomatically infected or if you have a very mild infection, that's enough for you to go out. And that's why these viruses are really good at community transmission, right?
I love this letter, and it's kind of, there's some layers to it. So the question I would have for you, DB, is if you had read that abstract or that paper, that Pain paper, before you got vaccinated, because then that would be the experiment, right? Is to give that paper as people are going in to get vaccinated, or maybe a day before, to half the group, and then the other half you don't give the paper, and then you ask afterwards, did you feel some buzziness or giddiness, some euphoria, some analgesia, perhaps, and then seeing if there's a difference between the groups. That would be kind of fun. But then I also, I want to hold out a little bit for this idea that, maybe not this, certainly maybe not this direct connection, but the idea of viruses themselves altering human or altering behavior of hosts. And there's some really good evidence. We've talked about this in insects, for example, some behavioral changes that like, the old story of the caterpillars getting infected with baculovirus, climbing to the tops of trees and exploding basically with virus particles that trickle down and then get spread to more caterpillars. And so, so it's a really interesting topic. Connecting that to human behavior is sort of like two orders of complexity, or layers of complexity on top of that, given our sort of complex behaviors and brains. But yeah, that's great. I love the idea, and some really interesting things there. Okay, next one. So this is from Kathy. Dr. LD, my dentist and I have had a long ongoing conversation about the disappearance of wisdom teeth. That is our first M1 molars in the human jaw. He explains that the loss is genetically driven to accommodate the decreasing size of our jaws due to change mastication and growing brain size. The question I have is, how is it that the genetic loss of wisdom teeth is asymmetrical? Many individuals only have 3 first molars and are missing 1 from either side. Doesn't the fetus start out as a flat pancake of sorts and curls together and binds down the center of the face. How does one lose the first molar and the other side retain it genetically? Thanks for clearing this evolutionary question, Kathy. Ooh, Kathy, this is a great one. I love this, and pulling us away from SARS-2 for a minute, which is a good thing too. But so this is kind of a developmental biology question, I'd say, and I'm going to just upfront plead a little bit of ignorance, except to say that asymmetry. There's a great literature, a great ongoing field on asymmetric. I'm getting asymptomatic and asymmetric kind of tangled in my brain. Asymmetric development, and some really interesting connections to the cell biology. So this has to do, for example, so we're not perfectly bilateral, right? We have two arms, but we have one heart that sort of sits a little bit to the side. And there's some really great work on how in early development, the cilia, the little hairs on the that sort of decorate some cells, the direction that they beat will move then molecules, developmental signaling molecules, to one side or another, and actually set up the symmetry or the asymmetry of the developmental system. And so this is totally speculating because I'm out of my depth here, but I wonder if that could be happening also in the sort of faciocranial regions and some of the sort of the asymmetric loss of molars of wisdom teeth.
I think we should do the last two and wrap up the bag, Nels. What do you think?
Yeah, that sounds great, Vincent.
So Ingrid writes, Dear Nels and Vincent, I don't manage to catch every single episode of TWiEVO, I'm afraid. My loss. So maybe you've covered or referred to this topic in an earlier episode. And Ingrid sends a link to a Quanta magazine article, which is loading. DNA of giant corpse flower parasite surprises biologists. And back to Ingrid, the genome of this plant is so weird that they can't figure out how or why it got that way. It's lost an amazing amount of important functional DNA, yet has a huge genome that is so repetitive and full of sequences kidnapped from long ago and now extinct hosts that it was very difficult to analyze. I doubt I will understand it even if and when you do go over the papers or the topic in general, but I'd love to listen. My husband and I used to be avid desert wannabe botanists out here in the desert areas of the western U.S., and there's an extremely tiny flowered parasitic plant, Pilostyles thurberi, that up to recently was included in the same plant family as corpse flower. Raphaelis, forget it. Rafflesia, SCA.
Yep,
so that anomaly is always mentioned in the few plant books that include this tiny parasitic plant, which is how we came to look up corpse flower. Sadly, to amateur wannabe botanists in the last 20 years or so, this tiny desert parasitic plants family has been split out of the Rafflesia. Its new family, Apodanthaceae, is even in a different order from the Malpighiales order that Rafflesia is in. Wiki claims Apodanthaceae is in Cucurbitales, but the Jepson manual says order attribution is still unresolved and could be either Malvales or Cucurbitales. The Jepson manual contains the definitive scientific descriptions of all California plant species, and the online version is constantly updated with the most recent scientific consensus. Popularly online and among amateur botanists, the two plants are still thought of as being closely related, one having the largest flower of any plant and the other close to the smallest. Either way, I would love to see someone figure out the genome of the tiny desert parasite and see if it is maybe even bigger than the giant corpse flower genome with similar anomalies, and she provides some links for this. Thanks for all the great TWiEVOs, TWiVs, TWiPs, immunes, etc. I love everything about the way you do these podcasts. So great to hang out with all you scientists and listen to real fun people talk about science. You've helped me get through this last year with its dearth of social interaction and reduction of classes, lectures, etc. But no, I'm not a temporary viewer just till the pandemic is over. We'll be relying on you guys for the duration, so please keep it going. Yeah, so we're here for the duration, so you don't have to worry about going away. Now, do you have any insight into this plant puzzle here?
Well, I was just thinking, I'm glad you read the letter with all of those scientific names. That was impressive, actually. So this is fascinating. So we haven't covered this on TWiEVO. This again highlights, I think both of us have a hunger to return to some non-SARS topics into the future. We're still on it for, I think some obvious reasons, especially as kind of controversies are emerging that overlap, I think, in pretty substantial ways with the evolution of the virus. But no, it's a great reminder, Ingrid, of how much incredibly cool biology there is around us. The corpse flowers. I mean, in addition to these crazy genomes that you're describing, also just the flowers themselves, right? These are just sort of these wild stories about natural selection, obviously on some really crazy traits in terms of flower, just morphology, but also the reason for calling them corpse flowers, the perfumes or the grotesque smells that emit from these, and the types of pollinators that they're animals that they're attracting. And so I have to confess, this is all pretty new to me, both the genomes and the certainly the kind of taxonomic relationships. But I think it is, and I would love to see more work in that area too. I think, as you're pointing out, when you have large genomes and repetitive DNA, that job becomes pretty tricky. But hopefully, there's some evolutionary geneticists out there working on it. I think the topics you're highlighting here are really fascinating. And
yeah, let's get one of these episodes in the future, Nels. That'd be great. One of these people who work on these plants would be great.
Totally. Okay, so our last letter of the day comes from Larry, Doctors Eldy and Racaniello. On the other hand, you've probably already seen this, in which case skip this letter. We haven't seen it, and so I think we saw it in the news, but we haven't covered it, or we've seen it in the literature. We haven't covered it on TWiEVO. So this is May 4th, links to 3 papers and 1 editorial in Science from the end of April, and some unusual bacteriophages, as the link indicates. So it turns out that the 1977 discovery of an alternate DNA coding scheme substituting 2-amino adenine, or Z, for the usual adenine, or A, and these are the 4 letters of the DNA A when we talk about ATs, Cs, and Gs. So instead of A, you have a modification to a Z, was not a fluke. There are more examples, and those are highlighted, as Larry's pointing out, in these recent papers that just dropped earlier this month. I believe you've commented recently on the difficulty of on how the difficulty of culturing in a lab affects what gets studied, and this is another example of that. Plus, Z, the base, is one of the amino acids found in meteorites, which should be the basis for a 1950s style science fiction film. And Larry proposes the title Bacteriophages from Space. No insightful questions from me, just wow, Z, T, C, and G. And would PCR be impeded by the appearance of Z instead of A? So we'd never know this kind of bacteriophage was present in samples. Thanks, as usual, for your truly interesting and informative discussions. This is from Larry, lifelong TWiEVO follower and software fanboy in San Diego. Larry, thanks. This is fun. So yeah, this is this really cool set of papers pointing out, or reviving actually, this idea of this alternate base Z instead of A as part of the genetic code. And so I actually you've put your finger on it. I have this bookmarked as a potential TWiEVO topic. Probably would have done it today if we weren't sort of still a little bit entangled in SARS-2 space. And so SARS-2 evolution. But yeah, this is totally fascinating. And I think you very nicely point out that how has this been sort of hiding under our noses, actually, or kind of, under the radar for so long. And I think in part it's true. We look at, as scientists, we look at such a narrow swath of the diversity of life around us for practical reasons. It's hard to study things that you can't keep in the lab, or that that are rare, or are in places that are hard to be with them, or to bring scientific equipment to them. And so, in addition to Z, there's probably, I can imagine, all kinds of fascinating variations on genetic code, other aspects of biology that are still waiting to be discovered. But yeah, this was a really fun story, and hopefully another one that we'll be returning to on future TWiEVO, or other discoveries like it, or advances involving that. And so for the PCR part, yeah, if you don't have Z in your collection of nucleotides, you're not going to see Z, because you just have adenine in there as you add that to the in vitro reaction. Yeah. All right, that's the mailbag. How about we wrap up with some science picks?
Sounds good.
I'll start. So, yeah, I think we're both going to hit on a few, I think, recent sightings in stories out there that caught our eye in the SARS-2 space that I think actually are kind of useful or positive in the conversation. So I have two. One from Ed Young, who's back. He was, I think, he was some incredible coverage of SARS-2 pandemic last year. I think he was working on a book, excuse me, and then now is back on the SARS-2 beat. And he has this latest one is on trauma, actually, from the pandemic, or as we step through it here now, 1 year plus on. And I found this to be a really nicely, not unusual for Ed, but a really nicely written sort of bigger picture view of some of the challenges that we might be facing, not just sort of virologically, but also psychologically as we continue to step through the pandemic. And I've got a link to that one if you haven't seen it yet. Second one from Yang Yang Cheng, and article from her titled on Vice. It's titled, "We're Not Ready for the Next Pandemic." And this actually does touch on some of the questions that came up from some of our live stream listeners. Some of the controversy or the ongoing, arguments about the origins of SARS-2. And I thought this piece, it's a long one, and I haven't, just from my scanning of it so far, I think actually kind of nicely sets the framework a little bit, gives a little bit of perspective on politics in China, some of the issues of transparency versus sort of how forthcoming you are with information. As Vincent is, and big recommendation to check out that as TWiV drops with this conversation about the World Health Organization ongoing investigations. I think this article really nicely kind of adds some perspective to the conversation. And so that's my second pick of the week here as well.
Very good.
Yep.
Excellent.
How about you, Vincent? Yeah. How about your science picks of the week?
Well, recently a very prominent virologist said that the SARS-CoV-2 furin cleavage site is a smoking gun for the lab origin of SARS-CoV-2. And, this was addressed a year ago on virological.org. And I don't understand why it's coming up again. Nevertheless, it's not a smoking gun. And my pick is a post by Christian Anderson, why it's not a smoking gun. And he goes through the very basic science, the molecular biology for why it is not. It's not, if you look at the protein sequence, you'll say, hey, it's not in closely related bat viruses. But if you look at the base sequence of the DNA, you can see that it's not just plopped there. Anyway, this is a good blog post. The fact is that other coronaviruses have furin cleavage sites. In fact, other bat SARS-CoV-2-like coronaviruses have parts of the furin cleavage site. And it's quite clear that it's out there and wasn't just put in. So Christian Anderson basically is repeating what was already posted a year ago on virological.org. But you know what? You can't say the right answer too many times, I guess.
That's right. Exactly.
We should check that out.
Speaking, Vincent, of this nameless but sort of famous scientific luminary that you are pointing to here, this is someone you're familiar with. Isn't this David Baltimore? And this was quoted
in fact,
by Nick Wade. I'm curious the backstory.
It's in the article. He's identified as David Baltimore, who I did a postdoc with, and who I have great respect for. Mm-hmm. I actually emailed him last week. I said, Dave, did you actually say this? And he sheepishly responded, yeah, I probably shouldn't have said smoking gun, but he said, Nicholas Wade kind of rearranged what I said. And I said, when I first saw it, it looked like, sticking out there. And so he said it, but I don't think he just hasn't looked into it. And here's the same problem we've been dealing with on this episode with the variants, with people not looking into the issue. If you're going to make a conclusion, look into the data before you do that, okay? And in this case, David didn't look at the data. It's very clear it's not a smoking gun. And I'm sorry, I feel very badly because I think he's brilliant. But it goes to show if you don't look at the data, and that's what I always say, I'm not by any means perfect, man. Forget it. But if you don't show me the data if you want to make a claim. If you want to say transmissible, show me the data. That's all I want. And if you have the data, I'll agree. And it's the same thing with the WHO committee. They said, if you have the data saying it's lab origin, give it to us. But nobody has the data. So that's unfortunately the backstory here. But, a lot of famous people make statements, and because they have a Nobel Prize, it is assumed that they're right. Luc Montagnier is in the press recently saying you shouldn't get vaccinated against COVID. It's a bad idea. And this is just wrong. And this can hurt people. This can kill people by saying things that are uninformed like that. So
yeah,
real problem.
No, thanks for that backstory. I was curious about that because I had read that article from Nick Wade, which I thought was a bit out of balance. And so that's, you're talking like a scientist, right? It doesn't matter that David Baltimore has a Nobel Prize. That doesn't mean that everything that he says will be on the money.
That's right.
And there are other Nobelists who are even, I think, way farther into the weeds on this in terms of some really potentially harmful takes on what's going on with SARS-2. Again, in these cases, no experience as a virologist, which is not the case for David Baltimore, but it highlights, of course, viruses are diverse, and our understanding of them is really specific to the features of them as well. But it's of course, for a number of reasons, a heated topic, and statements can get elevated pretty quickly in sometimes unfortunate ways.
Let me thank a few people here before we wrap up here. LWGSL, thank you for your donation.
Yes.
Susan K., thank you for your super chat. Very much appreciated. T. Klepper, thank you very much for your super chat.
Awesome.
I appreciate it. And I just want to say, we are open-minded here. We just are critical. Maybe that's the good word.
Well, and it's equal opportunity, right? I mean, you're talking about your postdoc mentor, and if that's something that doesn't line up with the evidence, you'll call that out. And so it's that's a scientific process or the scientific sort of philosophy or viewpoint that I'm
one
hearing.
More from JCU 2803. Thank you so much. We appreciate it. And
nice.
We'll be back next month, right, Nels?
I can't wait.
All right, that's TWiEVO number 68. You can find the show notes at microbe.tv/TWiEVO. If you want to send us a question or comment, TWiEVO@microbe.tv, or you can just come to the live stream and ask your question here. And if you like what we do, consider supporting us, microbe.tv/contribute. Nels LD is at cellvolution.org over on Twitter. He's L Earlybird. Thanks, Nels.
Hey, thank you, Vincent. Great discussion. I'm just getting a little bit more accustomed to this live streaming. I've been watching with great interest all of the comments in the site. I just wanted to throw out an appreciation to the conversation happening there, everyone tuning in real time, and for those donations, for that support. That's really a shot in the arm for us. So thank you all.
That's great. I'm Vincent Racaniello. You can find me at virology.blog. Music you hear on TWiEVO is by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.